Thank you for exploring the Research Information Center's Knowledge Base interview series. The Research Information Center is in the Florida Fish and Wildlife Conservation Commission's library, located at the Fish and Wildlife Research Institute in beautiful St. Petersburg, Florida. In 1955, the Fish and Wildlife Research Institute began as the Florida State Board of Conservation Marine Laboratory, with a mission to research red tide on a $15,000 budget. Today, the Fish and Wildlife Research Institute, a division of the Fish and Wildlife Conservation Commission, has an outstanding bank of knowledge in the retiring scientists who have spent decades at the Institute focused on a specific topic of great concern to Floridians. They leave behind a large collection of research valuable to future scientists who will build on their existing work. These research products are archived in the Research Information Center and available to the public through the FWC Digital Library. However, missing from these collections is the cache of knowledge that explains the scientists' relationship to their body of work, why they took a specific direction, and opportunities and limitations that shaped the research. The Research Information Center implemented the Knowledge Base interview series to capture this knowledge in recorded interviews of retiring scientists. We hope you enjoy their stories. Got a strange fish you cannot identify? Who do you call? Chief Ichthyologist for FWRI's Fisheries Independent Monitoring Program, Dr. Richard Ed Matheson Jr. That's who you call. The 2019 Florida American Fisheries Society Rich Kelter Award winner, Ed Matheson, has been a scientist at the Fish and Wildlife Research Institute since 1987. Dr. Matheson's research adventures include the effects of minimum flows on the Little Manatee River fish community structure. He investigated systematics and ecology of fishes, from freshwater streams to deep marine habitats. In addition to presenting at over 50 scientific meetings, Ed Matheson has authored over 30 peer-reviewed scientific papers that are listed at the end of this interview. We are sure you will enjoy the stories of this extraordinary man's career in marine research.
Good morning. We're here in St. Petersburg at the Florida Fish and Wildlife Research Institute talking with Dr. Richard Ed Matheson. Hi, Ed.
Hi, Adrian.
Thanks for joining us today to talk about your career here at the Institute.
Thank you. Thank you. It's a
How
pleasure.
are you today?
I'm good. I'm good.
Good, good. Well, I'll just dive in with a couple questions first.
Okay.
Let's start out with, could you let us know a little bit about your career path, what it took you to become a fisheries biologist?
Mm-hmm. Well, if you go way back, I always had aquariums when I was a little kid, and I always liked to fish with my father. And so when I got into college, I knew I was going to be in biology. And after that, there were some different paths I could have taken. I was offered to do graduate work on birds, but I didn't do that. I worked for a herpetologist, but he wanted me to do a fish project, so I did that at William & Mary, and then from that I moved on to do my PhD at Texas A&M, and from there I did a postdoc at Harbor Branch Institute on the east coast of Florida, and then another postdoc in New Jersey. I was all over the place, and then Mark Leiby actually sent a note to the person I was working with in New Jersey and said, "Do you know anybody that would be interested in a job in Florida?" Mm-hmm. And I applied for it and got it. So
great.
So that's how I ended up here.
So you've had experience in all different climates and places.
Oh yeah, yeah. You never want to sample in New Jersey in the winter. It's terrible.
All right, great. Well, that answers the next question. What or who brought you to work here? So I guess, what projects did you work on when you became a scientist here?
Well, when I first got here, I was working with Kevin Peters and Mark Leiby and also Behzad Mahmoudi doing early life history of mullet. Kevin was, we didn't have as much money back then. We were purely general revenue. There was no saltwater fishing license. And Kevin had been here for a long time already when I got here, and he was just in the habit of making his own sampling gear. So he made a channel net that we tried to catch baby mullet with out at Cross Bayou, and it was actually successful. And and he made cast nets, he made dip nets, and that was our first project, just trying to figure out where the early life stages of mullet were in the area and when.
Yeah. And what did you do from there? Did you?
Pretty soon after that, I got involved in the CZM study, which was a study funded by the Coastal Zone Management Program to look at fish distribution in the Little Manatee River in relation to salinity. There was a lot of concern about the effects of water withdrawal and stuff on tidal river fish communities, and that was considered to be the most pristine tidal river in the Tampa Bay system. Not pristine, but the most pristine in a relative sense. So they wanted a sort of a baseline study of that river.
Yeah. And then I understand for a long time you worked in the Fisheries Independent Monitoring Section as their chief ichthyologist?
Yeah, that was years later. There was a reorganization, and the old fish biology section that I was in got reorganized, and several of us got moved into different programs, and the FIM program, the Fisheries Independent Monitoring Program, needed someone to sort of oversee all the fish identification that they do because they sample constantly. And they knew I was good at that, so I moved over there. So.
Could you let us know a little bit about the process a taxonomist uses to distinguish species and larva stages from each other?
Right. Well, the first thing is you really need to know the literature, and there's a lot of it out there. There's a lot of species in Florida, and well over 1,000. Just in the area that FIM samples, there's well over 1,000. And there's literature going back to the 1800s that's useful. So the first thing you have to do is know all that literature, and then when you actually get a fish in front of you, you look at where its fins are placed on its body, you count the number of rays in those fins, you look at overall body shape. One thing that's very iffy is color. Some fish, like coral reef fish, you can only really reliably identify them based on color, but color doesn't last very long, either with frozen specimens or preserved specimens. So that's something you avoid if you can, but sometimes you can't avoid it. But those are the basic things: counting things and measuring things too. Body shape. A fish could be fat or skinny, but you actually can turn that into numbers, and that's what you do. That's called morphometrics, and you say the depth of the body is 40% of the length of the fish, and that will separate one species from another in some cases.
Wow. Very detailed analysis.
Yeah, it can take a while.
Yeah. Well, speaking of Fisheries Independent Monitoring, can you tell our audience what FIM's goal is and how IDing fish contributes to their mission?
Well, I should have the mission statement memorized, but I don't. But it's basically to provide timely data on the populations of fishes throughout Florida, mostly estuarine up until more recently they've moved to offshore also. But it's to provide timely data basically for the managers, and it was primarily focused on sport fish when the program began, things like red drum, because the managers wanted to know if the population was declining before they needed to do something. Like, if you can tell them that there are no 6-month-old red drum, they know that there's something wrong, and you can actually be more proactive in management. But then, since FIM actually measures, counts, and identifies everything they collect, the data can be used at a more community level too, like to see how the fish community is affected by things like freshwater inflow. And that's where you need to, most people can identify a red drum from this size up to that size, but not everybody can identify a mojarra at this size. And and they're very abundant. So that's where the fish identification becomes important, is mostly in those other species. Although I'll have to say that even with sport fish, sometimes it's easy to make mistakes. Like baby spotted sea trout aren't spotted. They've got a black stripe down the middle. And yes, we've seen people misidentify those because they got a black stripe instead of spots.
I think it's a snook or something.
Well, they usually think it's a freshwater fish because there's several freshwater species that have that black stripe.
So in addition to the community structure, that includes the predators and prey of a black drum. So if the prey is declining, you can say, well, black drum might be in trouble.
Yes, okay. You can get all of that from the FIM data. The FIM data, it's a stratified random sampling program. So they don't just go to a fixed site month after month after month. They've gridded off every estuary, and they're in estuaries throughout the state, and then random grid cells are taken, and that's where you're going to sample this month. But it's stratified because they force a certain number of grid cells to be in certain habitats like seagrass. So so it's a more statistically robust method than just doing fixed samples every month. Because basically with a fixed sample, all you know is what's happening at that spot. And you don't know if you're like really affecting that spot because you're pulling a net over it every month. You know?
So, true. Good point.
So they did do fixed station sampling at the beginning of the program, but they evolved to purely the stratified random.
Well, I'd like to ask you more about the Coastal Zone Management Project. Could you describe how salinity and habitat are impacted by changes in freshwater inflow, specifically the Little Manatee River where you started?
Well, one thing that people get wrong with the freshwater inflow issue is it's all about salinity, because certain species of fish do have salinity tolerances that prevent them from going in an area that's freshwater, say. But that's really not the biggest issue. The biggest issue is productivity, because without the water coming out of the watershed that's picked up all the nutrients from the forests and the marshes and everything, the productivity in the system goes way down. And you lose things, especially things in the base of the food chain like bay anchovies, which are easily the most abundant fish in Florida coastal waters, and everybody eats them. So if you lose that base of the food chain, you're going to have population declines even of things that don't care about the salinity. So it is salinity in a way, but it's also productivity, and that's what people have been worried about over the years.
Do you think that the work you did on that has impacted legislation or the way they restore tidal creeks or anything, like when the phosphate companies go in and try to?
Well, I hope it's used. I mean, we've certainly provided enough data, and the FIM program has done, that was just the Little Manatee. The FIM program has done similar studies in rivers all up the west coast, down to Charlotte Harbor, up to Weeki Wachee. And Tim McDonald has been the main person in charge of those. And I worked on a lot of them, and I don't know if you remember Marion Greenwood that was here. He was our resident Scottish person. But we did a bunch of reports on different river systems in Florida, all with the same goal of, how are changes in freshwater inflow going to affect the fish community.
Great. For our more non-scientific audience, could you please explain recruitment as it pertains to fisheries?
Well, yeah, it's one of those loaded words that really you got to be careful with the person you're dealing with and what they might mean. A real fishery person that's talking about, say, anglers and red drum, they'll usually use the word recruitment for things that are big enough to be caught by the fishermen. It's recruited to the fishery. But a more ecologically minded person might call a recruitment when the baby red drum come into the estuary. They've recruited to the estuary. So it's basically when they show up in something, whether it's in a fishery or in a habitat or.
Okay, that makes perfect sense. Thanks. Could you tell us a little bit about, I understand, I think it was in the 90s, there there was a phosphate acid spill over in the Alafaya River, and what might have happened when they tried to fix that?
Well, there's not much you can really do to fix that. But on the other hand, estuarine fishes are used to catastrophic changes, and they repopulate fairly fast as long as whatever happened to the water doesn't stay there. That the salinity or the pH in the Alafaya River dropped so dramatically that pretty much everything died. But it wasn't that long before things started moving back in, because that water, the new water coming in, diluted that, so that the pH went back up to safe levels and things started moving back in. So I don't know if the Alafaya is a very productive river. It's maybe the most productive river in the Tampa Bay system, and I don't know that you could actually tell that much difference now from the way it was before the spill. Now, right after the spill, there was a huge difference, but I think you could say that it recovered fairly well, because that's the nature of estuarine animals.
Yeah. I understand there was some kind of black drum habitat that might have been moved around. The people that were doing the restoration didn't realize that Bob McMichael and Kevin Peters had documented the most productive nursery habitat for black drum that anybody had ever discovered anywhere, as far as I could tell. Because I did a big literature review, so I don't think anybody had ever seen this many baby black drum recruiting. And it was a mud hole. It was ugly. And after the spill, there was money to do restoration work. And one of the and this wasn't that more recent spill. This was a previous spill in the Alafaya. They used that restoration money to restore this mud hole, and they turned it into a habitat that's really not very useful for baby black drum. They didn't know that, and it's certainly prettier. It looks nicer from above the ground, but not for the
right.
fish.
You just have to. If you're doing restoration work, you need to consult with a lot of people, local scientists, and read a lot of literature. And that just was an unfortunate one.
So the hydrologists and engineers would work together with a fisheries biologist to more adequately restore
and they usually do a really good job. That one's just one that didn't work out.
Oh, good. Good that they usually do a better job. Well, Floridians, one of the things we're talking a lot about recently is climate change and sea level rise. Do you have any theory or guess, or what do you think will happen to the tidal creek species when sea level rise comes in?
Yeah.
Or when there's even more population in Florida taking more fresh water for our human use?
Yeah. Well, that's two separate questions there, really. But well, let me back up a little bit. In all our work in the rivers, we noticed that we were sampling along the main stem of rivers, and we noticed that there were some little tiny creeks that were nowhere near as wide as this room, that had lots of fish in them, and specifically lots of baby snook and sometimes tarpon, which puts a dollar figure on it really quickly, since those are big game fish. But nobody was sampling those, and they didn't seem to be very protected. They would just disappear. Somebody built a trailer park, they would just drain it and fill it in. And so at one of the National Estuary Program meetings, they did one of those things where they put ideas up on a board and they get people to vote on them. And I said, maybe somebody should study the little tiny tidal tributaries in a place like Tampa Bay. And that's one of the things that got selected. So we ended up doing a bunch of projects, and that was a lot of that was Marion Greenwood. A bunch of projects on these little tiny tributaries that no one really cares about. And what climate change would mean for those is that they would become deeper water as the sea level rises, and at the upper end, they would still be theoretically at tiny tributary, but what if that upper end is now a parking lot? So, you know, we try to protect habitats that are really close to our rivers, but those won't be the critical habitats when the sea level rises. It'll be further back.
Yeah.
So they could run into a physical wall, really. As far as freshwater withdrawal due to increased human population, that will affect the productivity of the rivers, as I talked about earlier, and some of these tiny tributaries may just disappear altogether. So there's a lot of potential harm, and we know it's going to harm something. It's just hard to say exactly where it'll go, except we know if the water gets deeper, the reason they like those tiny areas is because they are somewhat protected from other predators. These are like little baby snook that are going to these places. There's not as many predators up there, supposedly, although birds are smart. They know where they are. But it's, you know, a tiny little creek. It's there aren't as many predators, and there's a lot of food because little live-bearing fishes are really abundant up there. And in the case of tarpon, they can even go beyond those little creeks into ponds that get anoxic seasonally because tarpon can breathe air, surface air. So you find them in ponds that have no dissolved oxygen in them. So they're the only game in town. I mean, they can hang out in there and eat everything, and nobody can bother them because nobody else can get up there.
Yeah.
So,
I understand you have a story about a little pond that was on Park Avenue up in
Park
Pinellas County.
Was it Park Boulevard or Park Avenue? It's a spillway going into I can't think of the name of that lake right now. But anyway, there's a creek coming. Well, there's a lake. There's a spillway. And then below the tailwaters below that spillway run into Cross Bayou. And Kevin Peters and I were doing our mullet exploration stuff in Cross Bayou, and we went up this little creek to the spillway, and at the base of the spillway was just this living soup of fish. And it was all these things that, in my interpretation at least, were trying to get upstream, but they couldn't go any further than that spillway. Except for the baby mullet, and I'm talking mullet much less than an inch long, there was a film of water that was really, really nothing at all on this spillway at an angle like that. And these baby mullet were climbing the spillway in hundreds. And nobody believes it, and we didn't have a video camera, but they were climbing it, and there were so many of them that big fat pelicans were actually raking their beaks along the spillway to get the mullet that were climbing the spillway.
It was a feast for the pelicans.
Except there's so little, they'd have to eat hundreds to get any kind of meal at all. But yeah, that was just, and the funny thing was there was a hipster-looking guy standing there, and he saw all the birds that were feeding on these fish, and he saw all the fish below the spillway, and he said, "Wow, man, it's really nice of the state of Florida to build this dam so the birds would have a place to feed." I didn't say anything. I'm thinking, I don't think that's why it's here.
All righty, let's see. How about over the course of your career, you've probably observed a lot of changes in habitats or technology, things like that. Are there any that stand out to you a lot?
Well, one thing that really stands out was technology, and it makes all the difference in the FIM program. I don't know how we did it in the early days, is GPS.
Yeah.
You know, when you've got to go to exact coordinates to sample at your assigned stations, I mean, it's very hard if you don't have GPS. And we used to have Loran was at the very beginning of my career, but then we had the uncorrected GPS there was something with the government had better satellites that we weren't allowed to use. I guess they were military satellites or something. And so the GPS that a member of the public could buy wasn't differentially corrected, so it could be pretty far off. And then we got programs that would actually do the corrections, and then we finally got GPS units that just did it all. You know, you just had to read the numbers off the thing. But that's been the biggest technological change. Otherwise, you know, the FIM programs use mullet boats forever, use the same seines and trawls forever. So none of that's changed. We're doing a lot more with video now, both inshore and offshore. So that's another, that's been around, but the video cameras haven't been as good as they are now, and they're getting better all the time, the underwater video.
They can even measure a fish underwater with
Yes,
the 2 laser
yes,
points instead
yes,
of having
yes,
a
they
diver
can.
go down there all the time.
Mm-hmm. Yeah,
Good.
yeah.
I bet that makes them happy in the winter.
Yeah, right. Yeah, well, with the CMAP program, the part that we're not the CMAP program, our offshore parts of FIM, they're measuring lots of fish that they just from videos. So that's, and it's good. It's totally non-invasive sampling. So
Great.
So it's good.
Where was your favorite place for field work?
Well, I really liked the little creek I worked in for my master's degree. It had a great name. It was Naked Creek, Virginia, and it was just a picturesque little creek. And I always say that's the last place I ever thought I understood the habitat I worked in, because it was so small. You could actually get a grasp on it. You know, it's hard to get a grasp on the Gulf of Mexico.
Yeah.
You know? But other than that, the Little Manatee River is really nice. It still is. It's changed some, but it's still I think the most pristine river system in the Tampa Bay area. Although, you know, there'll be development along it too.
I was there years ago. That reminds me I should go back again.
Yeah, no, it's a pretty river, and you can go to one of the canoe outposts and still have a fairly good wilderness experience, you know, going down the river. Yeah, for Florida. Yeah. Mm-hmm.
What was the biggest breakthrough or surprise of your career?
Well, a lot of things surprised me. Like when I was working on sculpins in the freshwater work, I was interested in comparative ecology, which is comparing the habitat use and diets and other things of species to see how they're sharing the resources, with the idea that if they were doing exactly the same thing, one of them's going to die out. You know? So I wanted to study a fish called a darter and a sculpin in the creek, in Naked Creek. Because I thought, well, they're pretty similar, you know, I wonder how they share the resources. And I talked to Bob Jenkins, who was writing a book on fishes of Virginia, and he said, well, you know, there's 2 species of sculpins there. And I said, no, I didn't know that. And he got me connected with the literature, and I found out, well, yeah, there are 2, and I found out how to separate them. I had to do that on my own because there was no good literature on that. And that ended up being my thesis. It was totally different than what I'd planned. It was comparative ecology of 2 sculpins. And then when I got started on my PhD, I was supposed to do an ecological project on these fish called mojaras, and there were 3 main species in the U.S. waters. There's some other minor species, but 3 main ones. And I started studying those, and I found out, yeah, they're really hard to tell apart. And there's not 3 of them; there's 4 of them. And it was, you know, I was able to document that very well. So my PhD dissertation turned into a taxonomic study instead of an ecological study, because it took me that long to figure those 4 species out.
So you discovered that one of the species was actually 2 separate?
Yes. And the standing joke was I took 3 species that nobody could identify and turned them into 4 species that nobody can identify.
Thanks, Ed.
Yeah, right. Yeah, everybody always loved it. And when I would do a talk at a national meeting, I would have pictures or slides of all the 4 species, and somebody would always come up. Some friend of mine would always come up after the talk and say, "You just showed the same slide 4 times. Those weren't there."
Great. Were there any collaborations with other agencies that really you worked with a lot?
Yeah, we've worked with a lot of different agencies over the years. Just here in Florida specifically, we did that thing with coastal zone management in the Little Manatee River. We've done a lot of work with the National Park Service down in Florida Bay and Everglades. And we were involved with the National Marine Fisheries Service and the U.S. Geological Survey and some of those things. Those are the main ones that I've been involved with where we were collaborating that I can think of right now. But those are good in a lot of ways. Funding, obviously, if you get different agencies involved, but also it broadens your horizons as far as contacts in the field and exchanging ideas. That's why it's good to go to scientific meetings, because it really helps. The networking in science is really important, and people underestimate how important it is, I think.
That's what I understood. Years ago, I talked to somebody who said, we just call up somebody in Miami and say, "Do you have this?" Because we all know each other. That's what's great about the community. Right,
yeah. And if you go to national meetings, you get to know all those people, and it's easier to make that phone call. You know, it's not a cold call. It's talking to somebody you've never met before.
Yeah, you put a face with a name.
Yeah.
Great. What advice would you offer to people who are trying to enter the fisheries biology field?
Well, what I usually tell students is, if you think you're going to make a lot of money, you need to go into some other field. If that's the only reason you're doing it, think of something else. But if you love it, then by all means do it. But that's got to be the main driving force, is that this is really what you want to do with your life. Because there are a few people that make a fair amount of money in our field, but not many. It's very rare. Most of us are pretty poor, but we love what we do. So, that old saying, "If you love what you do, you'll never work a day in your life." I think that's true.
Great. Are there any classes or skills that people should concentrate on that they might not think is involved in?
Well, a lot of people think that math is fairly painful, me included, but math, especially statistics, are very critical. So they need to just, bite the bullet and take those classes. I think an anatomy class is good, and that's another one where a lot of people have to bite the bullet because it's a lot of memorization. But then there are the standard classes, all the ecology classes, and something that gets ignored a lot nowadays is the taxonomy side. There are not a lot of good taxonomy programs based on morphology in universities anymore. Most of them are based on genetics, which genetics are great, but if you're doing something like a FIM program and you really can't run the genetics on 5 million bay anchovies, you need somebody that can actually identify them morphologically. And we aren't training enough of those people anymore. So basically the FIM people that come in, most of them learn their fish identification skills after they get here.
So that would be a great thing for somebody to have a niche saying, I'm one of the few people anymore who can do this. You're good.
Yeah.
Well, thank you. We really appreciate you
sharing
your time with us.
I enjoyed it.
I hope we all learned a lot. I sure did. And I'd like to just thank you for your service to the state of Florida, for your career too.
Oh, I enjoyed every minute of it.
Thanks.
Thank you for listening to the stories behind the research at the Florida Fish and Wildlife Research Institute. This video was produced by the Tampa Bay Library Consortium. For more videos from the Research Information Center Knowledge Base series, please visit TBLC's YouTube page and the FWC Digital Library website. To access the publications of this scientist, please visit the FWC Digital Library. To learn more about the Institute, visit the FWC website.